Welcome to this dynamic consent process. In this video, we provide you with the knowledge you need to have to understand the current situation and the reasons why additional research is needed. You selected that you have basic knowledge about DNA and genomic medicine. At the end of each video, we ask you if you understood the main points. If not, you will be redirected to a more exhaustive explanatory video. First, we will give you an overview of the history to emphasize that knowledge and research is constantly evolving. Second, we take a closer look at the human genome, what it is and what it does. And last, we shortly introduce the Swiss Whole Genome Project.
So, let's start with the history. From the antiquity already, human beings tried to find some theories to explain why there is a different species and what explains the resemblance between the generations. However, the greatest scientific advances have been made at the age of enlightenment. Among these, we mention a few. Pierre de Maupertuis, around 1700, wrote that modifications happens when two individuals reproduces, and this modification accumulates to create new races and species after enough generations. Georges-Louis Buffon thought that what we call species are in fact varieties that have been modified by environmental factors of the same species. Carl von Linné created the taxonomy of species that gather together similar species in different groups and subgroups, for example birds or mammals. Jean-Baptiste Lamarck proposed that the living beings become more complex over time because of some internal dynamics specific to their metabolism. Nevertheless, he also proposed that diversification of living beings is due to adaptation that are made by the individuals. The organs are kept and optimized if they are used. Otherwise, they atrophy over time. Finally, around 100 years after Pierre de Maupertuis, Charles Darwin explains in his book The Origin of Species his theory about evolution. The first detailed model of the DNA has been published in 1953 by James Watson and Francis Crick. So, what is DNA? DNA stores information by stringing together 4 different modules in a specific sequence. The human genomes consist of 3 billion of these modules. 99.9% of those 3 billion are the same in all humans, yet there are 3 million differences between humans. So your specific sequence is unique. Mechanistically, the very long strings of those 4 modules can be split in different entities. Some entities are like blueprints that tell your cells what it has to build. Those are called genes. Others are regulatory, that tell your cells how many and when it has to build something. Altogether, your genome determines many of your characteristics. Changes at a specific location, also called mutations, cause you to have lighter or darker hair, or may cause your cells to build something that makes you sick. Now, what does the Swiss Whole Genome Project? The first genome sequence was done between 1990 and 2003 with the Human Genome Project and cost around $3 billion. Nowadays, it costs around $1,000 and is done in some hours. These new technologies allow researchers to determine the sequence of many participants and then look for changes in the sequences that are shared between people suffering from the same disease. Today, however, we lack a large-scale database of genomes, which allows researchers to study still unanswered questions and make new discoveries. The Swiss Whole Genome Project aims at building such a collection of whole genomes.